Viking Mayer, and this is my oral communications project. Parkinson's disease is a neurodegenerative syndrome that affects dopaminergic neurons, leading to radical movements and tremors. The purpose of this paper, a Drosophila model of Parkinson's disease by Mel B. Fenney and Welcome W. Bender, is to test the alpha-synuclein gene in Drosophila as a comparable model to humans. Specifically tracking changes in dopaminergic neurons, filamentous intraneuronal inclusions containing alpha-synuclein, and locomotive dysfunction. Mutations in the alpha-synuclein gene have been linked to familial Parkinson's, and study of said gene can reveal underlying pathogenic mechanisms, thus making it possible to halt the degradation of Parkinson's disease altogether. The experiment seen in this figure was to show how degradation by alpha-synuclein was not limited to only the brain and could be replicated in the eye of Drosophila. What the figure demonstrates are the effects of wild-type and mutant alpha-synuclein expression during the first day of adulthood and day 30. It should be noted that these flies have a 60-day lifespan in which observation of the eye could have continued. Photos A and C are wild-type, and photos B and D are the result of mutant alpha-synuclein expression. The photos in the figure show a phenomenon described as retinal pseudopupils, which are extremely sensitive to changes in the retina structure. So what is observed is actually the degradation of the eye in terms of the conditions of the pseudopupils. The expression of both the wild-type and mutant alpha-synuclein gene during the development of the eye produced no effects, such as loss of eye or vestigial eye. However, as shown in the experiment, there is a clear degradation of the retinal pseudopupils due to the continued expression of the mutant alpha-synuclein gene, illustrated by pictures B and D. This change was detectable by day 10 in most cases and was formally reported on day 30, which indicates the modeling of effects in humans as well as in Drosophila. The states of the pseudopupils as compared in figures C and D provide clear evidence that mutated alpha-synuclein A30P or A53T rapidly degrades the structure of the eye. Fenney and Bender's conclusions based on the experiment suggest that this will facilitate generations of second sight modifiers and other manipulations designed to modify neurodegeneration. This is due to the presence of an easily assayed, nervous system-specific degenerative phenotype. What can also be interpreted is a relative rather than absolute specificity for the degradation of dopaminergic neurons. It is even suggested that because Drosophila produce more alpha-synuclein in the eye rather than the brain, it gives Drosophila resistance to alpha-synuclein toxicity. This experiment gives evidence that Fenney and Bender have created a successful model of Parkinson's in Drosophila by expressing human alpha-synuclein in the eye of Drosophila with results. They write, the parallel time course of dopaminergic cell degeneration, inclusion formation, and locomotor dysfunction indicates that the three abnormalities may be causally related. Page 4. The experiment of alpha-synuclein in the eye can then be related to other expressions as mentioned in the article, connecting all data to show a competent and accurate model of Parkinson's in Drosophila to that of humans, even going so far as to say Drosophila can provide a genetic complement to alpha-synuclein transgenic mice. This paper seems to offer hope into future studies on pathogenic mechanisms and novel proteins which may lessen the alpha-synuclein toxicity. The key point is looking into these routes as a way to lower the amount of mutant alpha-synuclein expression, similar to how we would want to lessen the number of genetic repeats when it comes to Huntington's disease, thus halting Parkinson's development or slowing down its progress. More generally, this can give ideas of how to study other neurodegenerative diseases by using Drosophila to study proteins and pathogenic mechanisms more rigorously.